Assalamu alaikum warahmatullahi ta'ala wabarakatuh. I'm Salma al-Mahdi and I will be your lecturer for this course titled Metabolism. We have certain objectives that we would like to meet by the end of this course, starting with outlining the main pathways of carbohydrate, lipid, amino acids, and nucleotide metabolism, to identify the main regulatory sites of these metabolic pathways, to identify the role of different tissues, including the liver, the adipose tissue, muscle, brain, RBCs, and to outline the principles of integration of these metabolic pathways, and to interpret certain pathophysiological changes with emphasis on the areas concerned with biomedical knowledge in relation to these pathways. And finally, to describe the principles of drug action in relation to certain metabolic pathways. This is a depiction of metabolic pathways. You can see that there is metabolism of carbohydrate right here, xenobiotics, other amino acids and other amino acids, energy metabolism, and so forth. It looks complex, but don't worry. We are going to tackle it one by one. We are going to concentrate on the main biomolecules. A nice analogy to metabolic pathways is just like traffic. Metabolic pathways, in metabolic pathways, we have molecules that can be analogous to various types of vehicles in traffic. We have pathways in metabolic pathways. In traffic, we have, of course, roads, streets, and railways, etc. We have networks of reactions comparable to maps and crossroads of traffic. And in metabolic pathways, we end up with end products or with the link to another pathway. In traffic, we have certain destinations that we would like to go to and certain stations where we would like to stop. In metabolic pathways, we have regulatory steps. In traffic, we have different traffic signals. And we have certain steps that can be rate limiting. In metabolic pathways, that is comparable to the traffic jams or bottleneck, where the traffic will be stopped or slowed down at certain points. And in metabolic pathways, we have the mechanism of feedback inhibition as a regulatory mechanism. Likewise, in traffic, we have automated traffic control, where we can monitor the traffic and we can control it live. Salvage pathways in metabolic pathways economically make use of certain shortcuts, just like detours or express highways or in traffic. In metabolic pathways, of course, you have abnormalities and unwanted products or unwanted reactions. In traffic, of course, accidents and can kill. So the analogy works at various levels. So how do we define metabolism? It is the interconversion of chemical compounds in the body. The pathway is taken by individual molecules, their interrelationship, and the mechanisms that regulate the flow of metabolites through the pathways. I would like you to make notice of the keywords in this definition. That is, we have interconversion, we have changing of chemical compounds from one form to another, and we have certain pathways, and there is interrelationship. So the pathways are not isolated. They are interconnected at certain points, and of course they are not random or haphazard. They are under control. So they are regulatory mechanisms that control the flow of those metabolites through the pathways. We have three categories of metabolic pathways. We have the anabolic pathways, in which there is synthesis of larger complex compounds from smaller precursors or building blocks. And we have the catabolic pathways, in which there is breakdown of larger molecules, commonly exotic pathways. Why? To produce reducing equivalents and energy in the form of ATP. And of course, we have a combination of both, that is amphipolic, which links anabolic and catabolic pathways, with a famous example of the tricyclic acid cycle. Normal metabolism in normal subjects may need to be adapted during periods of starvation or fasting during exercise, during physiological states such as pregnancy, lactation, and in growing children. Abnormal metabolism, however, results from nutritional deficiencies of biomolecules or of micronutrients such as vitamins or minerals, enzyme deficiency, abnormal secretion of hormones, whether they are increased or decreased, action of drugs and toxins, and disease states. All of these can lead to abnormal metabolism. So why do we eat? Of course, there will be different answers to this question. However, in biochemistry, and in particular in relation to the metabolism course, the answer should contain two main points, and that is to gain energy and to gain organic building blocks. Why do we need energy? To power living processes, such as synthetic processes. If you want to synthesize any molecule in your body, you will need energy. To contract your muscle in order to move, you will need energy. To transport ion across various channels to generate a nerve impulse, all these physiological processes require energy. And how do you get this energy? From food. Also, we eat to provide organic building blocks for the biosynthesis, of course, of larger, more complex structures. For example, we eat to obtain amino acids to build our own proteins for various roles of proteins in our body. Likewise, carbohydrates and lipids. In physiology course, of course, you came across basal metabolic rate and the metabolic rate. The metabolic rate, of course, is the amount of chemical energy liberated in the body per unit time, while the basal metabolic rate is the amount of energy expressed in calories that a person needs to keep the body functioning at rest. In the diagram to your right, you can see that boys and girls generally have a higher basal metabolic rates compared to adults, which is sort of a plateau. Has a plateau. Boys generally have slightly larger basal metabolic rate compared to girls, and of course we have the age in years across the basal metabolic rate in kilocal per meter per hour. That is the unit time. So what about the energy requirement? It is variable. According to what? To four main factors, including the physiological state. As we see, children requirements differ from adult requirements. Physical activity. If a person is leading an active lifestyle or a sedentary lifestyle. Hormonal levels, low versus high, for example, thyroid hormones. And mental state, manic, for example, versus depressed. A manic person will be very active, very energetic, so he or she will be consuming a lot of energy, while the depressed will have most likely will have low physical activity, low mental activity, and so on. So they are the opposite of each other. And you can see in the pie chart to your right that 60%, the green, represents the resting energy expenditure. So at rest, you are expending energy for the various physiological states that are going in your body. Synthetic processes, contraction. For example, if it's smooth muscles, your heart is beating, your intestine is moving, your brain and your neurons are firing up. So all these processes are consuming energy. In addition to the energy is expended on the physical activity shown in the in red, 32%. Digestion consumes about 8%. The energy requirement in the body is met by carbohydrates, 40 to 60%. Lipids meet 30 to 40%, proteins for 10 to 15%, in this order. So the main source of energy for our body is of course high carbohydrates, followed by lipids, and followed by proteins. Alcohol can also be a source, however, not a significant one for the humans. Variable oxidation depends on the duration and intensity of the physical work and the fed versus fasting states. So at any point of time, your duration and intensity of your physical activity, whether you are in the fed state or a fasting state, will determine your oxidation of these various energy sources. So, what is a fed state? A fed state is defined by two things: the time that elapsed since you last taken food, and the corresponding changes in the body energy processing. So, two things determine a certain fed state: the time elapsed since the last meal, and the corresponding changes in the energy processing in the body. And we have three fed states: the fit, the absorptive state, the fasting or the post-absorptive state, and starvation. And we can time them. The fed state is usually at 2 to 4 hours after a meal. A fasting post-absorptive state is late morning, late afternoon, or in the overnight, when there is no feeding for at least 4 hours or longer. And starvation, of course, if there is no feeding for continuous 24 hours. So, in the fed state, by definition, it is 2 to 4 hours after a meal. After 4 hours, the fasting state starts to up to less than 24 hours, after which, of course, the starvation state starts. In the fed state, there is ample supply of carbohydrates. So, the preferred metabolic fuel for most tissues will be glucose because it is abundant, it is available, so we can use it for energy. However, in the fasting state, glucose is limited, so it will be spared for the use by the central nervous system, by the neurons, and by the red blood cells. Fed state and fasting states, of course, differ, and they are almost opposite, in which metabolic pathways are active. For example, in the fed state, we have increased lipogenesis, we have active protein synthesis and active glycogenesis. So the synthetic processes for lipids, proteins, and carbohydrates are all active. And likewise, the matching hormones, insulin, will be high, while glucagon, the antagonistic hormone, will be low. In the fasting state, we have catabolic processes, so we have glycogenolysis, the breakdown of glycogen, proteolysis, the breakdown of proteins, lipolysis, the breakdown of lipids, gluconeogenesis, which is the synthesis of glucose from non-carbohydrate intermediates, and ketogenesis, which is synthesis of ketone bodies. All these processes mark the fasting state. The active hormone, of course, will be glucagon, while insulin will be inhibited. What about the balance of metabolic fuels? When the intake of metabolic fuel is more than the energy expenditure, so we have surplus energy, it will be stored in the body as triacylglycerol resource. At extremes, it will lead to obesity. On the other hand, when the intake of metabolic fuels is less than the energy expenditure, this will lead to exhaustion of the stores of carbohydrate and fats. They will be consumed, and following that, there will be mobilization of amino acids from the muscles to be used as energy source. We said carbohydrate and fats are the preferred fuels. When they are exhausted, the third option, of course, the body will look for another third source that will be muscles. They will be broken down to use the amino acids as energy sources, and at its extreme, this condition will lead to emaciation and wasting of muscle. And that is why, as a physical sign, certain muscles, particularly the facial muscle, temporal muscle, that's the ones that as the temple, if they are wasted, you will know that this person has been starved for a long time. To extend that, he or she has started to break down his muscles to provide energy. Metabolic fuels are different for different organs in the body. For example, the liver utilizes amino acids and fatty acids as fuel. It has the ability to store glycogen and some or little amount of triglycerides, because if the storage of fats in the liver has increased, you will get fatty liver, and that is a pathologic condition. So normally the stored lipid in the liver is very low. The fuel exported, so the liver can release fatty acids, glucose, and ketone bodies as fuels to the other tissues. The liver itself does not use ketone bodies as a fuel. However, it can export it. It can give it to other organs. We will see which one. Look here as the heart muscle, the middle of the table. The heart muscle utilizes fatty acids and ketone bodies in starvation. So during normal state, fed states, and post-absorptive states, the preferred fuel in the heart muscle will be fatty acids. If there is starvation, heart muscle will start to use ketone bodies as a fuel. From where does it get these ketone bodies? From the liver. It does not generate its energy. As you can see, the heart muscle does not store and does not export. So it does not store energy sources or fuels, and it does not synthesize, or it does not give other tissues. It is only a consumption state, in consumption state. Look at the end of the table. RBCs, they use the fuel utilized is glucose, the fuel stored is fuel exported is lactate. And we will see how glucose and lactate are related when we come to carbohydrate metabolism. So the only available form of fuel for RBCs is glucose. It doesn't have another alternative. So regarding the processing of metabolic fuels, the products of digestion are metabolized to a common precursor, usually acetyl-CoA, which will go into citric acid cycle. Look here, and this graph, we have food. It has been digested, absorbed, and then tested. In the bloodstream, we now have amino acids as products of digestion and absorption of proteins, glucose at the end, product of digestion and absorption of carbohydrate, and fatty acids as the end result of digestion and absorption of lipids, fatty acid of course, and glycerol. All of these metabolites will be processed into a common precursor, that is acetyl-CoA. Acetyl-CoA then goes in what we call the citric acid cycle. It is a cycle of reactions, 10 reactions. It is an amphibolic pathway. It links catabolic pathways like those of breaking down amino acids, glucose, and lipids to other synthetic pathways. The oxidation then will of acetyl-CoA through the citric acid cycle will generate reducing ethyl equivalent, represented here by the two hydrogens, that will go into what we call the electron transfer chain, and it eventually lead to ATP production. That is how food can produce energy, and we are going to discuss these pathways and processes in detail. We have a certain concept in metabolism, and that is compartmentalization, and that is to localize or to isolate a certain pathway in a certain subcellular compartment. For example, in the cytosol, certain processes that takes place in the cytosol include glycolysis, glycogenesis, glycogenolysis, pentose phosphate pathway, and lipogenesis. We have certain pathways that takes take place in the mitochondria, like citric acid cycle, beta oxidation of fatty acids, electron transport chain, and ketogenesis. In the endoplasmic reticulum, of course, we have triacylglycerols and protein synthesis as well as drug metabolism. The question is, what is the significance of knowing the specific location of a biochemical pathway? The first thing is that, for example, RBCs lack the mitochondria, so they don't have mitochondria. Eventually or consequently, they lack all the reactions that are related to the mitochondria. If you go back to the previous slide to show you, so RBCs cannot have tricyclic acid cycle. There is no beta oxidation, and there is no electron transport chain, and there is no ketogenesis in the RBCs. Why? Because they don't have mitochondria. So we will know this about RBCs, and this will help us in understand the metabolism of RBCs and the energy processing in that cell, and we will understand why blood cells depend on glucose as a sole source of energy. The understanding of compartmentalization is also important if you think of that molecules or products need to be moved from one side to another, for example, from where they are produced to where they are going to be consumed, whether it is inside the cell itself or outside, for example, to be secreted. And this, of course, will include crossing barriers like the blood-brain barriers, cell membranes, and so on. With certain molecules being hydrophilic and certain molecules being hydrophobic, and so on. Another important concept in metabolism is regulation or control. The flow of intermediates through metabolic pathways is controlled by four mechanisms, and of course you came across these when you studied the course of enzymes. The first one, of course, is the availability of substrate. Second one being allosteric regulation. Third one, covalent modification. And the fourth is a long-term mechanism of induction or repression of enzymes. Bioenergetics or biochemical thermodynamics are defined as the study of the energy changes accompanying biochemical reactions. When biochemical reactions take place, there will be an associated changes in the energy, whether it is consumption or production. The biologic systems, of course, conform to two laws of thermodynamic interactions. You remember delta G from your enzyme course. When a delta G, of course, is negative, that means the reaction will proceed spontaneously, like this one here. When it is less than 0, it will proceed spontaneously with loss of energy. It will be exergonic. Delta G is positive, that means the reaction proceeds only if free energy is to be gained, that is like going uphill. So it is this side of the diagram. It is endergonic. When a delta G is 0 at equilibrium, there is no net change of energy. And in metabolic reactions, we will come across various examples of different reactions that release energy or acquire energy. In metabolism,